Hi everyone, this is Nandini Shekhar. I'm pursuing second MSc Microbiology at Vivekananda Arts and Science College for Women, Sangatadi. Now, I will be presenting on the topic biological nitrogen fixation under the subject soil and environmental microbiology. This presentation has been carried out under the guidance of Dr. R. Dinesh Kumar Sir, Assistant Professor, Department of Microbiology. Before we get into the process of biological nitrogen fixation, let's first understand why nitrogen is so important. Nitrogen is one of the key nutrients that living organisms need for their growth and survival. For example, plants require nitrogen to make proteins, nucleic acids like DNA and RNA, enzymes, and even chlorophyll, which is essential for photosynthesis. Although nitrogen makes up about 78% of air around us, it exists mostly as nitrogen gas. In this form, it cannot be directly used by plants or most living organisms. That is exactly why the process of nitrogen fixation is necessary. It converts this unavailable nitrogen gas into a form that can be utilized for life process. Now let's move on to nitrogen fixation. Nitrogen fixation is basically the process of converting atmospheric nitrogen gas, which is not directly usable, into a form that plants and microbes can actually use, like ammonia and other nitrogen compounds. There are different types of nitrogen fixations. Broadly, it's divided into biological and non-biological. Biological nitrogen fixation is carried out by microorganisms, and this can further classified into symbiotic and non-symbiotic forms. Non-biological nitrogen fixation, on the other hand, happens through the natural physical process like lightning or even artificially through industrial methods. Okay, now let's move on to biological nitrogen fixation. Actually, we know that plants cannot use nitrogen directly from air, so some bacteria help them. These bacteria change nitrogen gas into ammonia, which plants can usually for growth. This process was first discovered by two scientists, Hermann Herschel and Martinus Beijering. Examples of such bacteria are Rhizobia, Azotobacter, Frankia, and Cyanobacteria. They use as a special enzyme called nitrogenase to do this job. So basically, these bacteria act like natural fertilizer of plants. Now let's look at the nitrogenase enzyme. This enzyme plays a central role in nitrogen fixation because it is the one that converts nitrogen gas into ammonia. It is actually made up of two parts, the iron protein and the molybdenum iron protein. The iron protein mainly supplies electrons, and molybdenum iron protein contains special cofactors called FEMO cofactor, which is iron molybdenum cofactor, which performs the actual conversion of nitrogen into ammonia. So overall, the nitrogenase enzymes works like a biological machine that makes nitrogen available in form that plants can use. Now let's see how root nodules are formed in leguminous plants. Root nodule formation starts when plants' root release flavonoids. Rhizobium bacteria sense this and release node factors. This causes the root hairs to curl and an infection thread is formed. Through this, bacteria enters the root cortex where nodules develop. Inside the nodules, bacteria turn into bacterioids and start fixing nitrogen for the plants. Now let's look at the different types of biological nitrogen fixation. There are mainly three types: symbiotic nitrogen fixation, asymbiotic nitrogen fixation, and associative nitrogen fixation. So these are the three major ways by which microorganisms fix nitrogen in nature. Symbiotic nitrogen fixation. Symbiotic nitrogen fixation happens through a mutual relationship between plants and bacteria. Here, plants provide shelter and some carbon compounds to the bacteria, and in return, the bacteria fix atmospheric nitrogen for the plant. This kind of association is long-term, ecological, and mutually beneficial. Examples of symbiotic nitrogen fixations can be grouped into three types: nitrogen fixation through nodules in leguminous plants, nitrogen fixation through nodules in non-leguminous plant, and nitrogen fixation without nodule. Now, let's see the first type of symbiotic nitrogen fixation, which is through nodal formation in leguminous plants. In legumes, the main nitrogen fixing bacteria are from the genus Rhizobium. These bacteria live inside root nodules. They can fix nitrogen only when they are present inside these nodules. The relationship is symbiotic because the plant provides food in forms of organic carbon, and in return, the bacteria supplies nitrogen to the plant. For example, Bradyrhizobium japonicum fixes nitrogen in soybeans and Azorhizobium colinodens forms stem nodules in Sesbania. So, this is how nitrogen fixation happens in leguminous plants. Next, let's see nitrogen fixation in non-leguminous plants. Some non-legumes also form nodules to form nitrogen. A well-known example is alder, especially in temperate regions. Here, the bacterium involved is Frankia, which is an actinomycete. This association is very efficient in fixing nitrogen and plays an important role in maintaining nitrogen balance in forest ecosystem and also in pure soils like glacial moraines. So, apart from legumes, even some non-leguminous plants can carry out nitrogen fixation with help of bacteria. Next, we have nitrogen fixation through non-nodulation. In this type, nitrogen fixation occurs without the formation of nodules. There are also called pseudosymbiotic associations. For example, we can see this in lichens where fungi live together with algae. It also occurs in sphyrobiotes with nostoc, in water fern azolla with anabina, and in cycads with anabina or nostoc. So, even without nodules, nitrogen fixation is possible through these associations. The next step is asymbiotic nitrogen fixation. Here, the organisms are free living nitrogen fixers. That means they can fix nitrogen on their own without depending on the host plants. They are found in both terrestrial and aquatic habitats. Now, different groups are involved: bacteria, fungi, blue-green algae. The first group is bacteria, which includes Azotobacter and Beijeringia. They are aerobic. Also, Clostridium, which is anaerobic. The next group is fungi like yeast and Frankia. And finally, blue-green algae such as Astroteria, a non-heterocystous algae, and Anabina or Nostoc, which are heterocystous. Now, let's move on to associate nitrogen fixation. Here, certain bacteria lives in close contact with the roots of certain crops and grasses, and they help in fixing nitrogen. This type of relationship is called associative symbiosis. The bacteria usually stay in the rhizosphere, that is the region between the soil and the roots, and sometimes they even enter to the roots. Part of the nitrogen fixed by them is absorbed by the plant roots. In return, the bacteria get food from the carbohydrates released by the root. Some common examples are Azoospermum, which is found with cereal roots, Beijeringia, which lives with sugarcane roots, and Azotobacter, which is associated with tropical grasses like Paspalum notatum. Let's take a look at the role of nitrogen fixation in the nitrogen cycle. The nitrogen cycle is simply a set of processes that change nitrogen into different forms in the environment. In this cycle, nitrogen fixation is very important. First, nitrogen fixation changes nitrogen gas into ammonia. Then, through nitrification, ammonia is converted into nitrates and later into nitrates. After that, in assimilation, plant absorb these nitrates for their growth. Finally, during denitrification, nitrogen is returned back to the atmosphere as nitrogen and the cycle is completed. Finally, let's look at the importance of nitrogen fixation. It helps to maintain the balance of nitrogen cycle. It naturally improves soil fertility and supports sustainable agriculture. It also reduces the need for chemical fertilizers, which can cause pollution and eutrophications. And overall, nitrogen fixation is essential for global food production. So, this is all about nitrogen fixation and its importance. Nitrogen may be invisible in the air, but its role in life is powerful. With that thought, I end my presentations. Thank you for listening. Bye bye.